Today I'll be talking about what happens during crossing over. Crossing over occurs between prophase 1 and metaphase 1, and it is the process where chromosomes pair up with each other and exchange different segments of their genetic material to form recombinant chromosomes. Independent assortment is the formation of random combinations of chromosomes in meiosis and of genes on different pairs of chromosomes.
I'm Brianna Chemist, and I will be discussing anaerobic respiration. Anaerobic respiration is the formation of ATP without the use of oxygen. Anaerobic respiration uses an electron transport chain, uses a final electron acceptor other than O2, which means in anaerobic respiration, oxygen is not used. It also is called fermentation and is used when there is not enough oxygen for aerobic respiration. Aerobic respiration is a respiration where oxygen is used. The equation for anaerobic respiration is glucose yields lactate acid and then energy is released. Basically, this equation means the breakdown of glucose leaves you with lactic acid and energy release. Pyruvates is basically the metabolization of glucose. Because anaerobic respiration uses no oxygen, pyruvates are then converted either to ethanol, which is alcohol, or lactic acid, which is used in the equation.
Today, I'm going to talk about what causes a disease like trisomy 21. We are living organisms that are made up of millions of cells, and inside the nucleus of each cell is genetic information found in chromosomes that determines a person's inherited features, which will be considered our DNA. So this is a picture of the chromosomes, and normally each cell that we have contains 23 chromosomes inherited from each parent, totaling 46 chromosomes per cell. The cell then divides so that all cells contain identical genetic information. Now, a person with Down syndrome has an additional chromosome 21. So, okay, here's where things go wrong. So, in this example here, as I told you before, normally each cell contains 23 chromosomes inherited from each parent. Now, a person with Down syndrome has an additional chromosome, which will be chromosome 21. So now you have 3 chromosomes instead of 2 on chromosome 21. And what happens is the breaking down and attaching to other chromosomes during cell division will have you with a chromosome with 3 chromosomes. And if you have this going on, then you will be diagnosed with Down syndrome. And this is why they call Down syndrome trisomy 21, because chromosome 21 has 3 chromosomes instead of 2. And that concludes my part of the research.
Hi, my name is Shekinah Strickland. I'm in Biology 121 with the Lab Rats. And my topic that I will be discussing is how meiosis is different in women and men. Is the purpose of meiosis? The purpose of meiosis is to reduce the normal diploid cell, which is 2 copies of each chromosome cell, to the haploid cells called gametes, which is 1 copy of each chromosome per cell. In humans, these special haploid cells are resulting from meiosis are eggs in females or sperm in males. Your diagram so that you can get a visual of what happens through the cycle of the male meiosis. The cell that undergoes meiosis in men is called spermatocyte. This cell makes sperm. It does meiosis 1 and it also does meiosis 2. The spermatocyte also produces 4 products of meiosis called spermatids. The spermatids aren't sperm because their tails haven't developed yet. In men, these changes begin to happen throughout the stages of puberty and continues throughout the male's life. The sperm begins to decrease when the male gets older. I'll explain to you about the male stages of meiosis. Right now, I'll be explaining to you about the female stages of meiosis. The female cell that undergoes meiosis is called the oocyte. The behavior of the chromosomes are similar, however, the cell divisions is different. Because the female needs well, the female egg needs to contain a lot of resources to support the growth of the embryo. All of these resources of the oocyte end up in the final cell, which is also the product of meiosis, called ovum. The ovum is a haploid cell that contains one replicated chromosome cell. The other product of meiosis are full-size cells. In meiosis 1, instead of producing two body cells, the other part of the cells that aren't wanted are formed, and they're called polar bodies. Meiosis 2 carry out another polar body formation. These polar bodies contain all of the chromosomes that aren't needed by the haploid ovum. Meiosis is more complicated in females. It starts sooner and ends sooner. It starts during fetal development. All of the oocytes that a female is going to produce are produced in the fetus. All of the oocytes begin to undergo meiosis 1. They start meiosis 1 in this stage part, while through meiosis 1, the chromosomes are lined up to separate, and they begin to pause. That pause is called a meiotic arrest. They hang in there between 10 to 15 years until the child reaches puberty. My name is Shekinah Strickland, and that concludes the research between male and female meiosis.
Hi, my name is Jasmine, and today I'll be talking to you about cell replication. When it comes to cell replication, the cell does a lot of things so they won't make extra cells. So for this demonstration, my keys will be the cell, and the door will be the G1 checkpoint, which is the G1 checkpoint is actually they make sure that all the cells are intact or they're the right shape and the right size. The G1 checkpoint that I will demonstrate later with keys and a lock is the beginning. It's the beginning of it all. And I say this because it checks the cell as the cell comes to the G1 checkpoint to see if the cell can divide and will it be a good working cell. The thing that makes it different, like when it comes to cancer, is that cancer is kind of tricky. We know cancer can end up later leading to lumps, which are normally tumors. Cancer is considered cells that wasn't supposed to copy when they did. Tumors are basically ungenerated cell divisions. So it just kept dividing and dividing and dividing. There are things called tumor suppressors that stop the cell from becoming cancerous. But as we know, if a cell is cancerous, it's malignant. If it is not cancerous, it is benign. For this demonstration, I'm going to use the keys to represent cells, and I'm going to use the lock to represent the G1 checkpoint. We know with the G1 checkpoint, it makes sure all the cells are the perfect shape and size before they go on to be replicated. First key, we're going to try the first cell. We're going to try the G1 checkpoint. It doesn't fit, so of course it won't unlock and it won't let it go through and be replicated. Second cell, we're going to try the G1 checkpoint. Like I said, it must be the perfect size and shape to get through to be replicated. It's the perfect size, but maybe it's not the perfect shape. So this particular cell won't be replicated. The third cell, we're gonna try this one. Perfect size. It unlocked the door. So which means it's the perfect size and shape. So this cell will be replicated.